These assays can be used to assess and compare immune outputs of different plant genotypes.
The main advantage of these techniques is that they are quick, reliable, and use common laboratory equipment.
For the growth inhibition assay, seedlings of the same size and age should be transplanted and fully blotted dry before weighing to limit variation.
At 4 to 5 weeks post-germination, use a sharp 4 mm biopsy punch to remove one leaf disc per Arabidopsis plant, avoiding the midvein and being careful to limit wounding. Collect the discs into individual wells of an unused 96-well luminometer plate containing 100 microliters of double distilled water per well, adaxial side up to prevent desiccation. If assessing multiple elicitors, remove a second leaf disc from the same leaf for each elicitor treatment, and cover the plate with the lid to allow the leaf discs to recover overnight at room temperature. The next morning, set the microplate reader parameters to a 1000 millisecond integration time in 2-minute intervals over a 40 to 60 minute period to capture the dynamic oxidative burst. Next, use a multi-channel pipette to replace the water in each well with 100 microliters of freshly prepared reaction solution, including a control no-elicitor reaction for each genotype, to assess the basal reaction oxygen species levels in the absence of elicitation. Then immediately measure the light emission for all of the wavelengths in the visible spectrum on the microplate reader. At 4 days post-germination, load each well of a 48-well plate with 500 microliters of MS medium supplemented with a dilution of elicitor peptide.